And so we move to our next speaker. Our next speaker is our Caparica talk of this year. Caparica talk tends to mimic the name of Caparica, okay, which is the place where we hold this conference. And normally we set one Caparica talk in every conference from someone who is doing something really new, or who is doing something which is not new, but from a new point of view, and which is something that needs to be highlighted in a science. We record the talk, and we make the talk available through YouTube and through our website, okay? And so once the talk is recorded and made available, I will send an email to all of you with a link. And this year, we have chosen one of the most promising researchers from Portugal, who is Rita Duarte. She comes from the Faculty of Science and Technology from the Nova University here in Portugal, here in Lisbon, here in Caparica. And she's going to delight us with a nice talk about natural deep eutectic systems and about the versatility of these systems in life science applications. Thank you very much, Duarte, for accepting to be here today. It's our great pleasure.
Thank you. So let me thank you for this introduction. Well, now the expectations are really high, so let's see how we manage to do this. I changed a little bit the title of my presentation, but it's more or less the same. What I'm going to talk about: natural deep eutectic systems in drug discovery and human health, breakthroughs in a liquid phase. And why do we call it in a liquid? Is a natural deep eutectic system. A natural deep eutectic system is a mixture of two or more components, which are typically solid at room temperature, but when they are combined at a particular composition, they become liquid. And this is what we see here in the images. Here we do have two solids, and at a certain point, they become liquid, so when combined at this particular composition. Here is what we see. We have crystals of menthol that when are combined with lauric acid, they become a clear liquid solution or a system. And these systems fully comply with the green chemistry matrix, which is something that is very, very interesting, and which is something that we don't see on the ionic liquids, which were the so-called alternative solvents for the future back in the 90s. So these systems, they don't have any chemical reaction involved. So the yield of production is 100%, and so is the atom economy and the carbon efficiency. In which concerns the E factor, which is a measure of the waste produced per product form, then this is virtually zero. And these systems have been named many, many different things. They have been named deep eutectic solvents or low transition temperature systems, among others. Well, we call them natural deep eutectic systems or therapeutic deep eutectic systems, as we are going to see in a minute. So natural deep eutectic systems were observed and are described in the literature since 2011, so not so long ago. And what scientists from Leiden University discovered is that these systems are essential in living beings for biosynthesis and biochemical processes. These systems are neither an aqueous nor a lipidic phase, but are a third kind of fluid phase that is present in the living beings and that enhances the solubility of some molecules and promotes chemical reactions of some of the processes that occur in these living beings. But what is this? How do they look like? Well, it's easy. We all have this in our kitchen. Like the honey, for example. Honey is a natural deep eutectic system, is a combination of sugars and also amino acids at different compositions. And this is also the reason why sometimes some honey crystallizes and others don't. It's because of their difference in composition. And in this particular case, the sugars have, provide the physicochemical properties, while the amino acids and phenolics contribute to the functional properties of these systems, like being antioxidant, antimicrobial, among others. Another example in nature is maple syrup. Maple syrup is produced in maple trees during a very short period of time, and it prevents ice from freezing in the trees, so that the trees can survive the low temperatures during the winter. And another example is taken from the sugar beet. The sugar beet syrup, which is used to de-ice the driveways in the Northern America. And sugar beet is a crop that is known to have a very high capacity to withstand severe environmental stresses. So both water dehydration and drought and dehydration. And interestingly, we see that in the shoot and in the root, so in different parts of the plant accumulates different components. Like, for example, in the root, we see an accumulation of proline, while in the root, we see an accumulation of sucrose together with betaine. And this prevents water from, or at least it keeps the plants from surviving under when water content level is very, very low. So the impact of these systems in chemical engineering and in chemistry is tremendous. They can replace volatile organic solvents. They can be used to design safer solvents and also to design less hazardous chemical synthesis. They have been reported in the literature to be used in biocatalysis, electrochemistry, extraction, even CO2 capture and biomedical applications. And the molecules that we study are primary metabolites, such as sugars, organic acids, or amino acids. But it gives us up to 10 to the power 6 possible combinations. This is really tremendous. So some of the examples that I'm going to show you here very briefly are related to the systems that we are studying in drug discovery and human health. We are testing these systems for their anti-inflammatory potential, antimicrobial, antifungal, anti-cancer, anti-tuberculosis, and tissue engineering. And we name them TDs, therapeutic dipeptidic systems. In these systems, at least one of the molecules that composes the system is an active pharmaceutical ingredient. And some examples perhaps are familiar to you, like, for example, these transdermal patches that are composed of flurbiprofen and menthol. These transdermal patches are patches that are put when you have a muscle contraction or something like that. And what happens is that the flurbiprofen is in a liquid state combined with menthol. Menthol helps the permeation of the anti-inflammatory agent, and therefore the activity is enhanced. And this is another interesting system which I have seen in the US, which is composed of choline salicylate and caffeine. And because if it's composed like this, it works, it claims that it works 5 times faster than aspirin in for example, for headaches and pain and muscle aches. Of course, this is because we have our molecule in the liquid state. This is what we try to do with the different components. So what happens now in the pharmaceutical industry is that the molecules that are discovered belong here to this class 4 of the biopharmaceutical classification system. So they have a low solubility and low permeability. And we want to tune them towards class 1 with high solubility and high permeability. And in the first ones that we discovered, we can see that we can, in fact, tune these 2 parameters, which may see exciting perspectives in pharmaceutical applications. The same happens for the design of antimicrobial systems. In this case, we move a little bit aside from antibiotics and we use fatty acids. Fatty acids are known to have these antimicrobial properties, and what we did was to combine several different fatty acids such that we can tune the melting temperature of these systems. And we prepared some gauzes, commercially available gauzes that are loaded with this system, and there's capric acid, myristic acid. This system is solid at room temperature, but when we place it on our skin, because we are nearly 37 degrees, this system becomes liquid and therefore it becomes more effective. And in fact, we made some antimicrobial and antifungal activity tests, and we do see that even if this case is combining menthol and fatty acid mixtures, we see that even if the system is not effective against the bacteria, it can help to detach the biofilm formed on a surface. So it means that, for example, if we coat a surface with this, a biomedical catheter, for example, the bacteria are not able to colonize these surfaces. And they are going to detach after some time of exposure. The same thing happens also for these fungi, Candida albicans, where we see that after 30 minutes of exposure to our DES, the bacteria start to detach as opposed to the control that we have here. And we have evaluated further these systems as regenerating agents in wound healing. And what we observe is that here we made a scratch in a cell monolayer, and what we observe is when we put our system in contact after 24 hours, and this is notable, after 24 hours we almost have full regeneration of the system. Of course, we can argue that this is similar to what we see with menthol, but the good thing about this is that we can complement this wound healing ability with the antimicrobial effect that we've seen before. So these systems have proven to be non-cytotoxic, have antibacterial activity, plus act as wound healing agents, enhancing 40% in comparison with the control. In terms of the tuberculosis treatment, the problem is that the patients need to have several drugs, and very long treatments are required. So what we aim to do is to combine these drugs to provide a more effective treatment to the patients. And we are still working on this, so we are working on these different molecules, trying to combine them in a liquid form to see if they are more bioactive towards the tuberculosis bacteria. This is still on early stage research. And another example is anti-cancer systems that we are working on. So here the idea is to use naturally occurring molecules, like for example limonene. Limonene has been reported in the literature to have anti-cancer activity. The problem with limonene is that it's not selective. So it kills both cancer cells and healthy cells. Plus, it's very volatile and therefore highly toxic. So it makes it difficult to work with. What we discovered here is that when we combine limonene with an anti-inflammatory agent, we can have enhanced properties of these systems. In particularly, we do see an enhancement of the solubility of the anti-inflammatory agent ibuprofen. We see a 4-fold increase in the solubility. We see that this system, when we combine limonene and ibuprofen, limonene is 4.4 times more selective towards cancer cell lines than when limonene is alone. And the system is six times less toxic towards healthy cell lines than the mixture, the physical mixture. So if we only combine the two molecules, limonene and ibuprofen, at exactly the same concentration, but they are not establishing interactions between the two, then the cytotoxicity effect is much different. The system has shown to have protective and anti-inflammatory activity as well as anti-cancer activity. And what we were talking about before, of course, a lot of work needs to be done in order to prove this and in order to move to the next stages of research. However, it's a first milestone that we achieved in order to continue the work that we are developing on this issue. In terms of perspectives, deep eutectic systems have numerous opportunities in different fields. I just briefly presented here some of the topics that we are working on relating on drug discovery and human health. And of course, besides this, we need to understand the mechanisms of action of these new liquid systems and to try to understand how do they interact with the cells. So finally, I just would like to share, to acknowledge the ones that I can share the excitement of every new experiment with, the researchers that are working with me in the lab, and also the financial support that allows us to continue this work. And thank you for your attention.
Yes, see. Yes, thank you very much, Ana Rita. Nice talk. So, I would like, as always, to begin asking some quick questions. The first one is, how do you realize that menthol was able to healing? That's really outstanding. And how really, so you have proved this in vitro, right? Mm-hmm. But how you try to test something in vivo?
Well, first the easy part, no, we didn't try any in vivo experiments because we are trying other combinations to see if we have better results than these ones. So we are first making a screening of different therapeutic systems, and the most promising ones we are going to pass to in vivo. So we still need to do a lot of work before going there. In which concerns menthol, I think there are already some works reported in the literature that that state that menthol has wound healing properties. So this is not actually something that is novel. The novelty comes from the fact that we make this new system in combination with the fatty acids and gives both properties, so the antimicrobial properties that are given by the fatty acids and the wound healing properties that are given by the menthol.
And what kind of bonds we're expecting to cure with this new combination of chemicals? So superficial bones, very
this
deep
is
bones?
This is for skin, so for superficial skins.
Okay.
Yeah.
There is questions in the audience? Yes, please.
I'm Yashoda from the Leibniz Institute in Germany. It was a very nice talk, and I really find this approach very innovative. I have a question which would probably help me understand, because I was thinking if you plan to put the eutectic mixture in the body, I would expect that the concentration is immediately diluted, and then how do you prevent the separation into the solid phase again, because the whole point is that you would like it to be a liquid so that it spreads quickly.
Yeah, so thank you for your question. The idea here is that we should look at this as, let's say, an API. And as all APIs, it needs to be formulated. So you need to find a formulation and a carrier, for example, for these. If we are thinking, for example, in the tuberculosis treatment, we are developing particles that can be inhaled and that go directly to the lungs, but we are not going to inhale the liquid form. So we can have a polymeric system, for example, and create a formulation before administration.
More questions? Yep.
Rita, congratulations, very nice talk. How combinatorial chemistry can help in your research in order to find new combinations of the different molecules? There are some kind of mechanical instrumentations that measure 2 or 3 components with the different quantities. How you can use these molecules is the first question. And the second one is about toxicity. The toxicity will be changed a lot depending on the concentration. Mm-hmm. Do you need to test any of the mixture after the end, or just the dose that the results are better?
I think combinatorial chemistry can do a great help for us, because at the moment we are doing a trial and error basis to test all these combinations. So to find the ratio at which the combination of the two molecules become liquid, it's done on a trial and error basis. There are already some thermodynamic models that can tell us more or less the affinity of the two molecules, but we still need to prove that by trial and error experiments. And yes, in terms of cytotoxicity, it's not linear what the outcome is going to be, because sometimes we see that there is a decrease when we combine this. For example, in the case of limonene, we see a decrease when it's combined with menthol, but for other molecules that doesn't happen. So still, we need to, for every system that we try, we need to test it.
Okay, if there is no more questions in the audience, we thanks Anna again.
Thank you.